Good evening, everybody. It's a pleasure to connect with all of you in today's webinar. So today's topic of the webinar is Klenza, a silver story, a promising antimicrobial in hygiene applications. Today's key spokesperson is Ms. VIVEGA Darshini, research scientist, RESIL Chemicals. We have panelist number one, Ms. RICHA AGARWAL, business development, Klenza, life sciences. Panelist number two, Mr. SIDDARTH, director, RESIL Chemicals. I thank Mr. SIDDARTH, Ms. Richa, and Ms. VIVEGA in joining in today's webinar. So the schedule of the webinar will start with a brief introduction about the parent company. This will be carried out by Ms. Richa Agarwal, followed by the presentation on the silver story by Ms. VIVEGA, followed by the question and answer session by Ms. VIVEGA and Mr. SIDDARTH. For any questions during the sessions, you can always raise the hand, which is you can see on the screen, and all the questions would be answered at the end of the presentation. You could also chat with each of the panelists post the presentation. So let's begin with the presentation. I request Ms. Richa to take over.
Thank you, Manoj. Hello, everybody. I, on behalf of Klenza Life Sciences and RESIL Chemicals, would like to welcome all of you for this first ever webinar of Klenza. To begin with, I'll like to share with you a short video on our company, RESIL Chemicals Private Limited. I hope the video is clear, and I'm going to start now.
Science is the fabric of everyday life. Be it the food we eat, the clothes we wear, the home we live in, or the car we drive, science touches every aspect of our lives. At RESIL, our story is about science. More specifically, a science for change. Founded in 1994, we began our journey by manufacturing silicones for textile finishing. Since our inception, we have been harnessing next-gen technologies to develop products that are high-performing, sustainable, and accessible. We pioneer scientific innovations and are driven by sustainable philosophy. Today, we are discovering science and its applications to meet the various technical requirements of the market. We are proud that leading global brands seek and rely on our acumen and capabilities in designing sustainable innovations. From chemicals for agriculture to antibacterials, healthcare products, and textile finishing agents, RESIL is at the forefront of sustainable innovation. Our global network continues to pioneer the inventions that lead to everyday solutions, enhancing the performance of thousands of products. We are powered by dedicated people who do what they do best by questioning the norm and reaching beyond the ordinary. At the core of our business lie four guiding principles: science, research, sustainability, and social responsibility, steering us in the pursuit of futuristic solutions. We are poised to meet the complex challenges of today and aim to create a better tomorrow. We are Science for Change.
So as you all saw in the video, RESIL has been a pioneer in the field of silicones and finishes from the last 25 years. In fact, the last 10 years has been really exciting because it's that time when we actually acquired the novel silver technology. And since then, we have won various prestigious awards and recognition. As you can see in the center, this award has been the highlight of all the awards because this was the National Technology Award given by the Honorable President of India, Dr. Pranab Mukherjee. I'll quickly facilities. Well, we have about 4 of them based out of Bangalore, Karnataka, India. And all of our facilities adhere to the global standards We have a capacity of around 20,000 metric tons and per annum. Now, moving on to the next segment of our webinar, which is the Klenza, the silver story. And to take you further to this exciting journey of silver, may I request Mr. VIVEGA to join the forum. Just to give you a quick background about Ms. VIVEGA Darshini, she is actually an MTech in nano silver and technology. She also has an extreme experience in the field of nano silver, antimicrobials, product development for hygiene applications, and I invite you to the forum to address the participants. Thank you.
Thank you, Richa. Ms. Viveka, you can start presenting now. I think audio is one. Yeah, we can hear you now.
Thank you, Richa, for the brief introduction. Good evening, everyone. Hope everyone is doing well. I would like to thank everyone for joining this webinar on Klenza, a silver story, a promising antimicrobial for hygiene applications. The purpose of this webinar is to demonstrate the benefits of silver for hygiene applications, its safety, and why to prefer silver technology over other antimicrobials. So this webinar will cover a wide range of topics. First, we start with brief introduction about Klenza. As this webinar will talk about the silver. First, we will start with a history of silver usage, its wide antimicrobial applications in various industries, and why silver technology is used in Klenza and its unique benefits. Next comes the very interesting topic, how silver kills bacteria and viruses. And we will see about specifically about the coronavirus also. Next, we will talk about the broad antimicrobial spectrum and how safe is the silver. Next, we will do the comparative analysis of silver versus other antimicrobials and why silver is preferred over other antimicrobials. So let's start the webinar. So it is the brief introduction about Klenza. So in a world where every touch is a risk we are taking, that too in pandemic, health and hygiene is the need of hour. So Klenza, a hygiene brand with wide variety of quality products, will protect our and our family from harmful infections and illnesses. It also meets the need of healthcare providers, food processing units, workplaces to reduce the spread of germs at their facility. Klenza range of products is formulated with goodness of silver, which will provide a better and safer protection without need of harmful chemicals like trichlorosills and parabens. Our first product is the Klenza alcohol-free hand sanitizer, which is India's first halal alcohol-free hand sanitizer, which will provide instant as well as over 3 hours germ protection. So we do have wide range of products like hand sanitizers, hand washers, wet wipes, Klenza mask sanitizers, masks, bed covers, stethoscope cleaners, and fabric sanitizers and travel-friendly surface sanitizers. So next, before moving on to deep into the silver technology, first we will see about the history of silver. So silver chemistry is not a brand new chemistry. It has been used in centuries and its antimicrobial properties are well known. So if we go back to 1500, acupuncture needles are crafted with silver, and in ancient times, silver vessels and silver coins has been used to preserve water, milk, vinegar, and wine. And Hippocrates, a father of modern medicine, has used silver preparations to treat ulcer. And in 17th century, silver has been used to treat cholera and epilepsy. Next, in 1920, US FDA has approved silver solutions to use as an antibacterial agents. In addition, silver preparation has been widely used for wound healing applications, silver fillings for teeth, and to prevent the blindness, and it has been used to treat the burn wounds. In second half of the 20th century, silver has been used for disinfectant purpose also. So far we have seen the history of silver usage. Now we will move on to how silver has been utilized in this modern era. So silver has been utilized in various fields. So an antimicrobial application of silver is highly valued in medical field. For example, it has been utilized for wound healing purpose, like it has been utilized for band-aids to promote the healing and also to prevent the reinfection. And in medical devices like catheter and bone implants, silver has been implanted in these devices to prevent the biofilm formation. And it has been utilized in textile applications, food packaging applications, and in animal husbandry. And it is also used to disinfect the various surfaces like surface, water, and air disinfectant. In particular, water and air filters are implanted with silver to impact the antimicrobial properties. And also in domestic products, it is highly utilized. For example, it has been utilized for in toothpaste, deodorants, filters, paints, and kitchen utensils. So this will give you the brief idea about few commercial products in the market. So for wound dressing applications, one product which is commercialized with silver particles, so which is the Acticone, which consists of silver dressings, which will give the broad bactericidal activities. Next comes the medical device, so which is a catheter, so which is incorporated with silver. Next, the silver gel, which is used for treatment of burn and wound infections, are formulated with nanocrystalline, and it is approved by the CDSC. So far, we have seen the history of silver and its current usage. So why silver technology has been used in Klenza? What is its benefit giving to the hygiene products? We will see. So first, why silver technology is used? There are so many benefits we will see. So first, it is the well-known and well-documented chemistry, which means it has been used in ancient times. So its antibacterial properties and its safety has been documented in various literatures and patents. And it has a broad antimicrobial spectrum, which means it kills bacteria, fungus, and virus. That too in a very low concentration. Even at a 1 to 50 ppm, silver is giving the greater than 99% microbial reduction. One unique point is it gives instant as well as the long-lasting protection. So it kills the microbes in less than 1 minute, and also it gives the residual, which means long-lasting protection to our hands for up to 3 hours. Next, it will work on the wide range of substrates. It can work on our skin and inanimate surfaces like hard and soft surfaces. Next, most importantly, it can be used on skin as well as the surface, which means it will not irritate our skin and it will not damage our surfaces. It will not leach, and it is non-flammable, and it doesn't have any volatile organic compounds. And our technology, silver technology, is patented, and one of our silver technology is US EPA listed. So these are the benefits associated in silver technology, which we are using in Klenza products. Next, we will move on into the how silver kills the bacteria. So before moving to that, first we will understand the basics about bacteria and its structure. This will help us to understand how silver works on the bacteria. So bacteria is a microscopic organism. Its size is very small. You can see it is 0.2 to 1.5 micrometer in diameter. So which is the oldest and structurally simplest form of life in the earth. It can present in everywhere. It can present in soil, water, air, and in our skin, in our gut, and it can present in the extreme environments like very hot temperatures also. So if we come to the structure, you can see the bacterial structure, which consists of several components. To make it easy, we can see the bacterial structure in 3 portions. So first portion is the structural components which are helpful for its movement and attachment. So the second component is the outer membrane. So third component is the inner membrane. So you can see the flagellum and phyllis. So these 2 components are helpful for cell mobility and attachment. Flagellum is a long hair-like structure which is helpful for cell mobility. You can see. And the small hair-like structures. So these are small hair-like structures, which is the phyllis. So this is useful for attachment to various environments. So the next comes to the outer region of the bacteria. So which cell wall. So this green portion is a cell wall. So cell wall is made up of peptidoglycan, This structure is the very important structure because it maintains the cell's integrity, bacteria's integrity, and it protects the bacteria from rupturing. So the next comes the plasma membrane. So below the cell wall, plasma membrane will be present, and it is responsible for, it is a permeability barrier. So it controls the movement of ions in and out of the bacteria. So the next comes the inner region. So you can see the yellow color thing. So it looks like the matrix, yellow color matrix. So above which so many intracellular structures will be embedded. So this blue color is the ribosomes. So it is an internal cellular structure which can be utilized for protein synthesis. So protein is an important biological component utilized in various biological functions. Next comes the nucleoid region. This purple color is the nucleoid region, which is nothing but a genetic material. Here in bacteria, it is a DNA. So genetic material can be DNA or RNA. Here it is a DNA. Next comes a mitochondria. Here it is not there. So many cellular structures will present. Mitochondria. So mitochondria, which is responsible for cellular respiration. So this is the basic structure of the bacteria. Next, we will move on to how silver technology works against bacteria. So before moving deep into the topic, first I will give you the brief idea about how silver works on the bacteria. So you can see the topmost right diagram. So you can see three set of diagrams. So first diagram. So in this first diagram, silver particles in area. So next, it will penetrate. It will idea. So now we will move on to the exact mechanisms by which silver kills the bacteria. So silver has multiple targets in the bacteria. So it can interact with multiple cellular components. So you can see the diagram. This yellow color is the bacterial structure. First, silver particles are positively charged, and bacterial cell membrane, which consists of negatively charged groups like carboxyl and phosphate groups. So this positive and negative will get attracted. So due to this electrostatic interaction, silver particles will get attracted to the bacterial cell membrane. You can see silver particles get attracted to the bacterial surface. So due to this interaction, cell membrane structure got damaged, and the internal cellular structures will get leaked. So due to this leakage, bacteria will die. So there is in one more mechanism. There are different mechanisms. The one more mechanism is these particles may penetrate inside the bacteria. So it can interact with multiple cellular components. So as I said, mitochondria will be present. So mitochondria plays an important role in cellular respiration. So once silver nanoparticles interact with the mitochondria, cellular respiration will get affected, due to which bacteria may die. Otherwise, it may interact with the proteins. So proteins plays an important role in various biological functions, and so if silver nanoparticles interact with the proteins, it got denatured. So due to this dysfunction, bacterial dysfunction will happen and it will die. Otherwise, it will interact with the genetic material, which is a DNA. So due to this interaction, DNA is required for replication. So once silver nanoparticles interact with DNA, bacteria will not replicate and it will die. So the next mechanism is production of reactive oxygen species. Silver nanoparticle will produce the reactive oxygen species inside the bacteria. So due to this, stress will be created inside the bacteria. So due to this stress, proteins and lipids got oxidized, and these will be involved in the respiratory process. So due to this, cellular respiration will get affected and it will die. So the next, the last is signaling pathway may got affected. So silver nanoparticles will interact with the signaling pathway, which are responsible for the bacterial growth. So it will hinder the signaling responsible for the bacterial growth, and finally bacteria will not multiply and it will die. So these are the mechanisms by which silver will kill the bacteria. Next, we will move on to how silver kills the virus. So before going to that, we will understand the basics about the virus and its structure. So virus is a obligate intracellular parasite, which means it requires host to multiply. Without that, it will not multiply. It will be metabolically inert. So without host, it will not multiply. So virus structure consists of two components. One is nucleic acid, another one is capsid. So you can see the structure, generalized structure of virus. So here you can see the two structures. So the first is a naked virus, which means the inside red color thing is a nucleic acid. Nucleic acid is nothing but DNA or RNA, which is a genetic material. So the outermost thing is the capsid. So capsid is nothing but made up of protein coat. Which means it covers the nucleic acid. Basically, it is a protective coat for this nucleic acid. Next, pure virus will consist of one more structure, which is called envelope. You can see the enveloped virus. This yellow color. It is the envelope which is made up of lipids and proteins. So this in this envelope, proteins will be projected from these surfaces. So this is called a spike. So, virus may present in different shapes, polyhedral, helical, spherical. So, kind of its capsid structure may present in different structures. So, you can see the naked virus and enveloped virus. The virus which we have recently encountered is the enveloped virus, which means it has additional outer coat. So, we will see about the coronavirus also. In the late 2019, a novel coronavirus outbreak has been happened, and most of them who developed the pneumonia kind of symptoms. So the virus has been identified as a coronavirus family, and it has been named as SARS-CoV-2, and the associated disease is called COVID-19. So as I said, the SARS-CoV-2 belongs to the coronavirus family. So the name comes after it looked like a crown-like spikes on their surface. You can see the blue color and the purple projection. So these are the spikes present in the coronavirus. So we will see its structural components. So here also you can visualize this virus in the three parts. One is the outer spikes, and the next is the outer membrane. Next is the inner part. So the outer spikes is responsible for binding with the host cell receptor. So this spikes plays a major role to entry into the host cells. Next comes the membrane, the outer shell. So the outer shell will consist of different proteins, which is the membrane glycoprotein. This membrane protein is required for the structural integrity. Next comes the inner part. So inner part will consist of the RNA. So coronavirus will consist of RNA as a genetic material, and protein will be embedded. So this is the basic structures about the coronavirus. Next, we will see how silver technology works against virus. So silver particles is strong antiviral agent. So there are two different ways silver will work against the viruses. So first we will see the left side diagram. Here you can see the two portions. So first you can see the rightmost portion. So how virus infection happens, and the leftmost portion how silver affect this viral infection. So first we will understand how virus affect our host cell. So this is the virus particles, and it will interact with the host cell, and it will release only its genetic material. In case of coronavirus, it will release only its RNA. So inside the host cell, it will multiply using a host cell mechanism, and it will kill the host cell and it will be released. So this is the basic viral infection mechanism. So how silver prevents this is silver interact with the viral particles, virus outer surfaces. Due to this interaction, virus particles will not interact with the host cell. So you can see, so here it can't interact with the host cell. So due to this, viral entry into the host cell will be blocked. So the next mechanism is this silver particles will interact with the genetic material. So I told then genetic material can be RNA or DNA. So this silver particles will interact with the viral genetic material. So due to this, viral replication will be inhibited. This is the two mechanism. First, it will hinder the attachment with the host cell. Next, it will interact with the genetic material and it inhibits the replication. This is the two main mechanisms. So you can see the rightmost diagram, so which gives the broad antiviral activity of silver. So it has silver particles will work on wide range of virus. So HIV virus, HBV virus, hepatitis B virus, herpes simplex virus, influenza virus. So these are the wide range of virus against which silver will work against. Next, so far we have seen the mechanism by which silver works against the bacteria and virus. Now we will see about how our silver technology has been tested and how we have found the broad antimicrobial activity of our silver. So all our Klenza products has been tested as per the international standard, which is the ASTM standard, against wide range of microbes, bacteria, fungus and virus. And we have found it will give greater than 99% microbial protection in less than 1 minute, as well as it is giving a long-lasting protection up to 3 hours. So I have told it will work against bacteria, fungus and virus. There are different fungal species, bacteria species, virus species. So next we will see against what are all the bacteria our silver has been tested and we have found efficacy. So this slide will give you an idea. So against what are all the test organisms we have tested and we have found the efficacy. So in the left table, you can see we have tested against gram-positive as well as the gram-negative bacteria. So these are the test microorganisms, Staphylococcus aureus, Bacillus subtilis, E. coli. So these are the test organisms. And these organisms will cause wide range of human infections. It will cause urinary tract infections. It will cause health care associated infection. It will contaminate the food. It will cause a pneumonia. It will cause diarrhea. So these are the various infections these bacteria will cause, against which our silver has been tested and it is found to be efficient. So this bacterial efficacy has been tested as per ASTM standard. Next, antifungal efficacy. So this antifungal efficacy has been tested as per the ASTM standard. We have tested two fungal strains, which is the Aspergillus niger and Candida albicans, which will also cause fungal ear infections, and Candida albicans will cause urinary tract infections. And we have tested one of the viral strain, which is the MS2 bacteriophagy. This is basically a virus which will infect the bacteria E. coli. So we have tested our silver technology against this virus. And we have tested one of our silver technology against coronavirus also for medical textile applications. So far we have seen how silver is effective against microbes. So next comes the important topic, how safe is the silver? So our silver technology has been studied for different toxicology studies. So first we have done an acute oral toxicity study. So from this we have found its LD50 value is greater than 5000 milligram per kg. So which is categorized under a category 5. So basically as per GHS, category 1 is the very harmful and category 5 is less harmful. So category 5, from this we found silver technology which we are using is less harmful. And we have done the skin irritation study. So and from this study we have found it is non-irritant. And we have done an eye irritation study. From this we have found it is not irritant. And we have done the skin sensitization study. And from this study we have found it is non-sensitized. Next, we have done the 28 days repeated dose dermal toxicity study. And from this, we have found its NaOEL is greater than 1000 micrometer per animal. Next, we studied our silver technology to test whether it is mutagenic. So from these two studies, it has been found it is non-mutagenic. So overall, from this toxicology study, we found it is non-irritant for skin and non-irritant for eye, and it will not sensitize our skin and it is non-mutagenic. Next, we will go on to why to prefer silver technology over other antimicrobials. So here we have taken a wide range of antimicrobials used in most of the commercial products. So first is a chlorine compound. So chlorine is a halogen-based biocide, and even a lesser dosage, even in a ppm level, it will work. But the disadvantage is it will not give a residual efficacy because it is unstable, it disappears quickly. It has, even though it has a broad antimicrobial spectrum, like it will work on bacteria, fungus, virus, etc. It is corrosive to eyes and skin, and it is a respiratory irritant. And also, it will damage these surfaces. It will corrode the metals. It will bleach the fabrics. It will damage the fluids. And it is a strong oxidizer. More importantly, it will release the toxic chlorine gas when mixed with ammonia or acid, and it will produce the carcinogen upon contact with formaldehyde. So, even though it is it gives a wide antimicrobial activity, so it has so many disadvantages, like it is corrosive in nature to our skin and to the surfaces also, and it will produce the possible carcinogen. Next comes the hydrogen peroxide. Hydrogen peroxide, as we know, it is a strong oxidizer, and at 3% it will act as a disinfectant. And for this also there is low residual efficacy, and it has a good antimicrobial efficacy. But it is a serious eye irritant like chlorine compound. It is also a corrosive, and it is not compatible with substrates like zinc, copper, nickel, and it will decolorize the fabrics also. And at high concentrations, it will give this sharp odor. Next comes the iodine compounds. So iodine compounds, even at a less dosage, it will work fine. Again, it will not give a residual efficacy. Sometimes for these iodine compounds, bacteria, virus, it will work, but it requires prolonged contact time to kill the fungus and spores. Next, it is also a skin and eye irritant, and it is also corrosive. It will stain, more importantly, it will stain the surfaces like plastics and pores. And it is non-flammable, yes, and it will be less effective at a lesser pH. Next comes the benzalkonium fluoride, which is a quaternary ammonium compound. So it is effective at a less dosage, and it will give the residual efficacy. But it has a bactericidal fungicidal efficacy, but it is not effective against the non-enveloped viruses, and it will cause a contact dermatitis and respiratory sensitizers, and it is slightly corrosive, and it is associated with the antimicrobial resistance, and it is easily inactivated by the presence of organic matter. So next comes the alcohol. So alcohol, a very high concentration is required to achieve the required efficacy. 70 to 85% is required. And it is work against the wide range of microbes, but it will not destroy the spores. And the risk is it is poison upon injection, and it will cause eye irritant, and it will cause skin irritation. So it is non-corrosive, but it is flammable. The disadvantage is solution is not effective at a lesser concentration, and it will evaporate easily, which will not give a residual efficacy. So by comparing all these things, if we go to the silver technology, even at the lesser concentrations, even 2 to 5 ppm is effective in a lesser contact time, and it gives the residual efficacy. And it is effective against wide range of microbes. And as per our toxicology study, it is found to be non-irritant and non-mutagenic. And it is non-corrosive. And it is safe on skin also, and it is safe on wide range of surfaces. And it is non-flammable. So it is its non-flammable nature. It doesn't have volatile organic compound. It is safe for skin. It is safe for substrates. So this technology will provide better user experience compared to other antimicrobials. So these are the cleanser products which has been formulated with silver technology. So we do have a wide range of products. If we go with hand hygiene, we do have alcohol-free hand sanitizer, alcohol-based hand sanitizers, and hand washes. If it is for hygiene essentials, we have silver mask and shield sanitizers, and wet wipes. And for surface hygiene, we have surface sanitizers, seat brush, which is nothing but a toilet seat sanitizers, and stethoscope cleaner. So we do have 2 more products in pipeline, which is Sanidio, which is nothing but a fabric sanitizer, and Travel Guard, which is a travel-friendly surface sanitizer. So these are the products Klenza have. Next, we move on to question and answer.
Thank you, Ms. Viveka, for such an extensive insights about silver. We now move on to the question and answer session. So the forum is open for questions from all the attendees. You can raise hands from the button that you see on your right hand of the screen. You could also chat with individual panelists if you have any questions. Also, the questions could also be vocally asked in the forum. Once a participant raises their hand, we can unmute them, and then you can put forward your question and the questions will be answered. All right, we have one question from Namaste Enterprises. Good evening, sir. So you're unmuted now. You can ask your question. All right. Meanwhile, while we figure out the issue with Namaste Enterprises, we have another person who has another question. Mr. Bhupesh, you can put forward your question now. You're unmuted. You can ask your question now.
Yes. Good evening, everybody. This is the
Good
question.
evening, Mr. Bhupesh.
Yeah, this is a question to Viveka, madam.
Oh, yes, sir.
Madam, is our silver technology will rest? How long it takes to destroy the bacterial spores, ma'am?
Bacterial spores.
Yes.
Yes, sir. So it is effective against bacterial spores like bacillus and Clostridium species. So as per the literature, it is effective in a 5 minutes.
How long it takes, ma'am? 5 minutes?
5 minutes. Yes, sir.
Madam, can you explain about the difference between the sterilization and disinfectant, ma'am? If you don't mind.
Yes. So disinfection is almost a killing of bacteria, virus, and fungal strains up to 99.999%, greater than 4 log reduction. Sterilization, which includes the removal of spores.
Yeah, that is what, ma'am. Sterilization takes more time.
Yes.
Usually, what happens first? If liquid will disinfect first. Suppose our liquid is taking 1 hour time for sterilization. Within the period of first it disinfects the surface. It takes another half an hour or 20 minutes to destroy the bacterial spores.
Okay.
So then only it considered as a sterilization, right?
Yes, sir.
So that is what I am asking, because you have not mentioned in our column the comparison thing, which we don't that bacterial spores. Particularly, it was not mentioned.
Yes, sir. Yes,
that is what I was.
Yes, as we haven't tested specifically for the spores, I haven't mentioned, but as per the literature, silver technology is effective against spores. So that is what I told. It is effective against bacillus and Clostridium spores, and it is in 5 to 10 minutes.
Right, ma'am. Because we need the why? Because mainly the bacterial spores is found in only hospitals related at very sensitive areas only, like operation, which it can't be found in other areas. In my experience, I think it takes more time for to destroy the bacterial spores. Then only it considered to be. That is what I'm asking you. Anyway, thanks for your answer. Thank you.
Thank you, Mr. Bhupesh.
Thank you. Thank you.
Yeah. I think the next question is from Mr. Abdullah. His question is, will the presentation be available? Yes, the contents of the webinar would be available. It will be sent to you post 24 hours post the webinar. It will be sent to you through an email. All right. I guess there are no more questions from the audience. Let's move on to the next part of the webinar. I now request Mr. Siddharth to give us a closing note.
Right. Thank you, Manoj. Thank you, VIVEGA, for that fantastic presentation. I feel like I have learned a lot. In spite of being part of the hygiene and business, I feel like I have learned a lot. So thank you for that fantastic information. I would like to thank all our participants and our valued customers and partners who have joined this webinar. We really appreciate your commitment and your passion towards making Klenza the leading hygiene brand in your regions and your segments. Klenza was founded on the principle that there should be better forms of hygiene which do not harm the user and should also be comfortable for the user. We do believe that people require safer, more comfortable, hygienic solutions going forward. Silver being the absolute backbone of what we do. Silver is a very interesting technology. Every day it makes us, as we work with this technology more and more, we find it more and more remarkable. We find it amazing. We find it to be very incredible in terms of its, and very powerful in terms of its applications. So we are very happy to ensure that more products can be made along with silver. We also do work with some other active ingredients, but we do believe silver is the best. And we thank you for working closely with us on this. There is a question from Mr. From Namaste Enterprises. Sorry, sir, I don't have your name. The product, your question is, our Klenza products are FDA approved. So in India, the approval authority is Director General of Cosmetic of DGCI, Director General for Drug Controller Authority of India. My mistake. So that's the main approval, and we are DGCI approved. We are also approved in the Philippines as well, thanks to one of our partners who's also there on this call, Mr. Ishuji. He's been very instrumental in helping us get that. So yes, we are approved. There are a lot of products that do not require approval. For example, the mass sanitizer, which is a surface application, which does not require approval. So the products that require approval, like the hand wash, the hand sanitizer, they're all approved. I hope, sir, this answers. So thank you, everybody, for joining us. We really appreciate all your passion and your commitment. And I look forward to meeting all of you personally when it is safe. Meanwhile, stay safe, stay hygienic, and let's work together to make Klenza the leading brand of hygiene in 2021. Thank you.
Thank you, Mr. Siddharth. I once again thank all the attendees of the webinar and the panelists, Mr. Siddharth, Ms. Richa Agarwal, and the key spokesperson, Ms. VIVEGA Darshini. I look forward to meet you all in the upcoming webinars of Klenza. The contents of the webinar would be shared through an email shortly. Thank you all.